Hey everyone, welcome back to the channel. Today we are diving into a fascinating topic biology for engineers. If you think biology is just for doctors and scientists, think again. Engineers are now using biology to design better materials, improve healthcare, and even build living machines. Why engineers should care about biology? That is your question. Engineering has been all about physics, maths, and chemistry, but the 21st century is all about bioengineering. From biomechanics in prosthetics to bioinformatics in AI driven healthcare, biology is reshaping engineering fields. Earth is unique among known planets because it has the perfect conditions to support life. Here are the key factors that make the earth special. There is presence of liquid water. There is moderate temperature range on the earth. The free oxygen in the atmosphere makes the earth very unique. We have plenty sunlight. There are no toxic substances in the atmosphere. There is absence of lethal radiation. Yes, the earth is very hot. The energy from colliding meteoroids heated its surface. Earth's early atmosphere probably contained hydrogen cyanide, carbon dioxide, carbon monoxide, nitrogen, hydrogen sulfide, and water. Due to this, the volcanic activity formed on the earth. It resulted in releasing of great amounts of water vapor that later condensed to form oceans. The released carbon dioxide that warms the atmosphere by absorbing outgoing heat. This early atmosphere lacked oxygen, so it could not support life as we know it today. You might have plenty of questions. Where did life begin? How did life begin? What did it look like? There is fossil evidence which indicates how the life began. The fossil evidence indicates that life on Earth appeared about 3.5 billion years ago in the oceans. The primordial soup theory is a widely known hypothesis about the origin of life on Earth. It suggests that life began in a soup of organic molecules that formed in Earth's early oceans through chemical reactions driven by natural energy resources. This theory proposes that simple organic molecules like amino acids and nucleotides formed spontaneously in Earth's early environment. Over time, these molecules combined to create more complex structures, eventually leading to the first living cells. Miller-Urey experiment tested the primordial soup theory. The scientists Stanley Miller and Harold Urey in 1953 conducted a famous experiment. They simulated early Earth's conditions in a closed system containing water, methane, ammonia, and hydrogen. They introduced electrical sparks to mimic lightning. After a few days, they found amino acids, the building blocks of life, that had been formed in this experiment. This experiment provided the first experimental support for the idea that life's essential molecules could have arisen naturally. There is a difference between the abiogenesis and biogenesis. Abiogenesis or spontaneous generation states that life can arise from non living things. Apparent reason that oxygen prevents the synthesis of certain organic compounds that are necessary building blocks for the evolution of life. Oparin proposed that the spontaneous generation of life did in fact occur once, but was now impossible because the conditions found on the early Earth had changed. Oparin argued that a primeval soup of organic molecules could be created in an oxygen-less atmosphere, whereas biogenesis states that every living thing came from a pre-existing living thing. In 1668, Francisco Redi proved that no maggots appeared in meat when flies were prevented from laying eggs. In 1768, Lazzaro Spallanzani demonstrated that microbes were present in the air and could be killed by boiling. In 1861, Louis Pasteur performed a series of experiments which demonstrated that organisms such as bacteria and fungi do not spontaneously appear in sterile and nutrient rich media. There are certain criticisms and there are certain alternative theories. While the primordial soup theory is popular, there are other competing ideas such as hydrothermal vent hypothesis. According to this theory, life may have started at deep sea vents where hot, mineral rich water provided the necessary conditions. Another theory is panspermia. Life's building blocks may have come from space via meteorites. So, clay may have provided the foundation for first organic compounds. Mineral crystals in clay could have arranged organic compounds into organized patterns. The ice earth. 3 billion years ago, ice might have covered the oceans. It got protected from UV light. The organic compounds may have formed and reacted with one another. How did cells form? Sidney Fox and other scientists have done extensive research on the physical structures that may have given rise to the first cells. Cell like structures, including microspheres and coaggregates, form spontaneously in certain kinds of solutions. Coaggregates and microspheres, like cells, can take up certain substances from their surroundings. Coaggregates can grow. Microspheres can bud to form smaller microspheres. However, microspheres and coaggregates do not have all of the properties of life. Unlike cells, microspheres and coaggregates do not have hereditary material. When did atmospheric oxygen appear? About 2.7 billion years ago, cyanobacteria or blue-green algae began photosynthetic reactions. About 1.8 billion years ago, the atmosphere contained abundant free oxygen. It allowed for the development of more complex oxygen breathing life forms. It caused the first mass extinction of organisms that had evolved in an oxygenless planet. Spirulina is a type of blue-green algae that grows in both freshwater and saltwater. It is one of the most nutrient-dense foods on the planet and has been consumed for centuries due to its amazing health benefits. It has high protein, which is rich in vitamins and minerals. The endosymbiotic theory explains how eukaryotic cells, that is, the cells with a nucleus, evolved from simpler prokaryotic cells. Prokaryotic cells are nothing but bacteria like cells. It suggests that key organelles like mitochondria and chloroplasts were once free living bacteria that were engulfed by a larger host cell. Instead of being digested, they formed a symbiotic relationship, eventually becoming a permanent part of the cell. What are aerobic prokaryotes? An aerobic prokaryote is a single celled organism that requires oxygen to survive and generate energy. These bacteria and archaea use oxygen in cellular respiration to produce ATP, the energy currency of the cell. Examples of aerobic prokaryotes includes Mycobacterium tuberculosis, which causes the tuberculosis and which thrives in oxygen-rich environments like the lungs. Nitrosomonas, which helps in nitrogen cycling by oxidizing ammonia into nitrate. Thus, these aerobic prokaryotes are very important because they are very essential for ecosystems. They play key roles in carbon, nitrogen, and sulfur cycles. They have medical significance. Some cause diseases, while others are used in antibiotics. They help in bioremediation. Anaerobic eukaryotes are single celled or multicellular organisms that can survive and grow without oxygen. Unlike most eukaryotes, which rely on oxygen for energy, these organisms use anaerobic respiration or fermentation to generate ATP. How do anaerobic eukaryotes generate energy? Since they do not use oxygen, they rely on fermentation. They convert glucose into ATP without oxygen, producing byproducts like lactic acid or ethanol. These anaerobic eukaryotes rely on anaerobic respiration. They use other molecules like sulfate or nitrate instead of oxygen to generate energy. The examples of anaerobic eukaryotes includes Giardia lamblia, a parasite that infects the intestines and survives in low oxygen environments. Trichomonas vaginalis, which is a sexually transmitted protozoan with hydrogenosomes instead of mitochondria. Entamoeba histolytica, which causes amoebic dysentery, thriving in oxygen poor intestines. Loricifera, which is a tiny marine organism that live permanently without oxygen, which is found in deep sea sediments. The main key steps in endosymbiosis includes a larger primitive cell engulfed smaller bacteria. Instead of digesting it, the two cells started working together in a mutualistic relationship. Over millions of years, the bacteria became a permanent part of the host cell, evolving into modern day mitochondria in animal and plant cells and as chloroplasts in plant cells. Both mitochondria and chloroplasts have their own DNA, which is similar to bacterial DNA. Both mitochondria and chloroplasts can replicate independently. They divide like bacteria through binary fission and separate from the cell's nucleus. Both mitochondria and chloroplasts have double membrane. Their inner membrane resembles bacterial membranes. The ribosomes resemble bacterial ribosomes. The ribosomes produce proteins which look like bacterial proteins. Endosymbiosis is very important because it explains the origin of complex life. Without this process, eukaryotic cells would not exist. Endosymbiosis became key to evolution. This allowed cells to become more energy efficient, leading to multicellular organisms. మర్చిపోకుండా సబ్స్క్రైబ్ చేసుకోండి.